reproduction, and life cycle in mucor. Mucor is a very common fungi belonging to class Zygomycetes. It is found to be growing on a wide variety of substratum. That is, it grows on soil with a wide range of pH, which means that it is able to grow both under alkaline as well as acidic condition. It is also found to be growing on organic substratum like dung, cow dung or horse dung, on decaying fruits and vegetables, on bread. In fact, mucor is very common bread mold growing along with rhizopus, which is another zygomycetes, and another two fungi, that is aspergillus and penicillium. Though it has been found to be growing along with rhizopus, penicillium, and aspergillus on the same bread, but in fact, the vegetative growth is fine. But when it comes to reproduction, that is during the sporulation, these fungi may be inhibitory to one another. For example, the toxin, mycotoxin, rubatoxin that is produced by Penicillium rubrum, it is found to inhibit and totally suppress the sporulation in mucor. So we see that these fungi, these four fungi, they are growing together, but when it comes to reproduction, they are inhibitory to one another. And in fact, the sporulation in mucor has also been suppressed by certain fungicides or fungistatic chemicals such as thymol and the captafol, terazole. These all, they also have very uh, suppressive action on the spores of mucor. Apart from that, natural products like any soil also is very much repressive on the sporulation of mucor. Hello friends, I am Dr. Ayantika, and in this video I am going to discuss about the reproduction and life cycle. So reproduction in mucor, it occurs by asexual as well as sexual method. Asexually, they produce two types of spores. Under favorable conditions, sporangiospore are produced, and under unfavorable condition, chlamydospores are produced. And in fact, the sexual reproduction, it occurs. These are the chlamydospore that are produced under unfavorable conditions. So, the asexual reproduction, it occurs at much relatively unfavorable condition, which means that during relatively unfavorable condition of a little amount of drought or water stress or high temperature, this type of asexual reproduction occur. And sexual reproduction occur when the conditions are very conductive, because if there is drought stress, then the negative strain, we know that mucor, it is heterothallic and have got two type of strains, positive and negative strain, and a presence of these both strains are very necessary for the sexual reproduction to happen. So, in any case, if the drought stress occur, then the negative strain, it loses its capacity to form the gametangia, and that is why during unfavorable condition, asexual reproduction occur. But in asexual reproduction also, when the water condition is water stress is too much high, then we have chlamydospore instead of sporangiospore. So first we'll be discussing the sporangium or the sporangiospore. We see that mucor, rhizopus, and absidia, these all produce sporangium. Well, the structure and the formation of sporangiospore is similar, That is, they are produced in cluster. These sporangiospores, they are produced in cluster from the horizontally growing aerial hypha, but there is a bit of structural difference. That is, in rhizopas and absidia, the mycelium the mycelium or the vegetative body is divided into this stolon, rhizoid, and the sporangiospore. But in case of mucor, this type of rhizoid or the stolon or the mycelium, this type of division, it doesn't occur. The same mycelium, it penetrates on the substratum, and this mycelium is just a mycelium but not much highly branched rhizoid. So this is a basic vegetative structure difference between mucor and rhizopus. Otherwise, the sporangiospores, they are produced almost in a similar way. So this sporangiospore, they develop as an aerial branch. They develop as an aerial branch from this vertical aerial branch from this aerial hyphae. So this vertical branch, they arise in cluster, and this branch is called the sporangiophore, and the tip of which is called the columella, and this is the sporangium. So let's discuss this in detail in the next slide. So when there is an aerial mycelium, erect hypha, sporangiophore, they arise. The tip of this sporangiophore, they swell. The tip swells and accumulate high amount of nuclear and all the reserve stored matter. And the central region of this tip, this central region becomes highly vacuolated, and later the vacuoles they fuse to form a continuous layer that separate the central vacuolated portion, that is this portion, from the outer portion, that is this one. This outer portion, which is called the sporangium, produces the spores. This outer portion has got a high density of nucleus, high density of reserved food matter, and gradually the protoplasmic content of the sporangium, it gets separated to form the multinucleate mass called the spore. And these each of this multinucleate mass, they get surrounded by a wall which is two layered, and this outer electron dense and inner less electron dense, and it has got all the spores, they have got multi nuclei and all the organelle. So this is how basically the sporangium is formed. So this is the stalk, the sporangios. This is the sporangiophore. The stalk is called the sporangiophore, and this is the columella. This is the sporangiophore, the stalk-like portion, the central portion that is surrounded by the sporangiospore. This one is the columella, the central region, and the outside region is called the sporangiophore, each of which contains spores. So one thing is that the wall of this sporangium, it becomes thick and encrusted with various granules. And in fact, when these spores, when they mature, the sporangial wall dissolves. Now, once the sporangial wall dissolves, what happens is that the sporangial wall is absorbed, and only a little portion of the sporangial wall, it remains in this stalk region as a collarette. Collarette. So the earlier in rhizopus we have seen that the wall of the rhizopus, it dehisces, and all the spores, they are liberated by air current. But in case of this is in the case of rhizopus. But in case of the mucor, what happens is that the sporangial wall, they dissolve and remain attached as a colorate, and the spores that is present, they are usually not liberated by and dispersed by wind current, but they are liberated by, they are dispersed by mite. Those certain portion, they are also um, they are also dispersed by wind, they are also dispersed by wind, but mostly they are dispersed by mite. So the spores, these spores, they when they fall on suitable substratum that is containing high amount of sugary material or starchy material and high amount of water, that is high water activity, then only they will germinate and they will produce a new mycelium. So, the asexual reproduction by which the spores are produced, there the haploid generation is multiplied. The haploid generation is multiplied. And asexual reproduction by production of spore is a very common method. Then uh, there are certain conditions that affect the sporulation. In the first slide, I talked about the presence of certain fungicides, certain fungistatic substances, or toxins from other type of fungi. Those they inhibit the development of spore. But development of spore is also affected by certain conditions like temperature, light. We know that usually the fungi, mucor, it grows from 5 to 25 degrees C, but it can also grow at 37 up to 37 degrees C. But at 37 degrees C, we do not see much sporulation. And also at very low temperature, like 15 degrees C, the we have the sporangia formed, but they have a structural difference. That is, at this 15 degrees C, the sporangiophores are very short. But on the other hand, if it grows on 20 degrees C, then the sporangiophore are tall. This is the sporangiophore structure. So, based on the temperature, this sporangiophore height, it it is alters. And then we have light. When the mucor is growing under dark condition, it will grow because it does not need light for its food production, because it is a saprophytic organism. So it produces exogenous enzyme, dissolves the starchy material that is present on the substratum. So it does not require light for its growth, vegetative growth. But of course, for the reproduction, it requires light, because uh, light alters the the length of the sporangiophores, because in very dark condition, short sporangiophores are produced mostly. And what happens when short sporangiophores are produced? The spores that is produced inside the sporangium will not be dispersed to far off places, because wind current, because if the sporangiospore is at very low level, then the wind current will not be able to lift it and take it to further places. So this is one reason. These are the terms while the sporulation is getting affected. So this mucor, it has got certain positive as well as negative relation with other type of fungi. The mucor is getting hyperparasitized by these type of fungi, and once it is hyperparasitized by these type of fungi, then the sporulation or sexual reproduction is totally suppressed. However, this mucor can also it badly affects human as well as animal by increasing the immune response, especially once the complement system of our adaptive immunity it is getting activated, then we have several type of inflammatory response. So this mucor, though in very low quantity and under normal condition will not be affecting us, but if we are already sick or we are immunocompromised, then this is a very good opportunistic organism. It produces the spores inside our body, and entire body gets infected, and the infection spreads. And if this type of mucormycosis or zygomycosis is not treated, then the spore, they actually damage several of our organs, and then these spores are responsible for our death if we are not treated. So the spores of mucor is very damaging under immunocompromised person. So another type of spore, that is the chlamydospore, that is produced under extreme drought condition. Basically, these chlamydospores are produced when the mycelium, it it accumulates several protoplasmic content at intermediate region, and these, I'm drawing it in a different color. So these intermediate protoplasmic region, they have dense cytoplasm containing high amount of reserve food matter and many nuclei. And in fact, these region, they get a wall. This accumulated protoplasm get a very thick wall. And the intermediate region that I'm drawing in green, so these intermediate region, they are empty. And this chlamydospore, they are so much resistant to drought that when the substratum, under extreme drought conditions, substratum dries up, entire mycelium dies, but this chlamydospore will survive. And on return of suitable condition, that is on return of moisture, they will again germinate to produce the mycelium. One thing maybe I forgot, that this asexual spore, that is the sporangiospore, that also germinate, produce a mycelium. So this is about the chlamydospore. Chlamydospore is a method of perennation or to tide over the extreme drought condition in mucor. Then we have sexual reproduction. So we already know that mucor is heterothallic. And as because it is heterothallic, which means that it will have two mating strains. Now these mating strains, they may be physiologically, they may be morphology, morphology of this opposite strain, they may be same. But physiology, there is different. Because different because physiologically it is different, because they are genetically different. They produce different type of proteins and all that help in mutual attraction. So these two type of strains, that is the positive and negative strain, and in fact when the opposite strained mycelium is close enough, then they produce trisporic acid, that is a attractant. And in fact, only if this trisporic acid is present, then only the mycelium, they form this progammetangia type of structure. So this is the progammetangia. Progammetangia. So what happens is that this is the mycelium. This is the mycelium of a positive let me draw it here. This is the mycelium of a positive strain, and this is another mycelium of the negative strain. So this is positive, this is negative. So only when they are close enough, they develop certain special branches towards each other. And for the development of this entire thing, that is development of the spatial branches called progammetangia, trisporic acid is very much essential. It acts as a stimulant. So once this progammetangia is formed, the tip of this progammetangia, it undergoes swelling, Once the swelling occurs, then what happened? They've come close to one another. They come close to one another, and a septum is formed. A septum is formed, and this septum actually, they actually divide the progametangia into this gametangia and the suspensor cell or the basal cell. So, once this progametangia is formed, and so this is the gametangial portion that I'm drawing in green, and let me draw it in a separate color. So this is the suspensor cells or the basal cells, and this is the gametangia. The region in contact, the wall in contact of the two gametangia, they when this gametangia is mature enough, they dissolve. And once they dissolve, then plasmogamy occurs first, which means that the protoplasmic content of this gametangia, say this is the positive strain, this is the negative strain. So protoplasmic content, they mix and they form this structure called the zygospore. Zygospore, and this zygospore, after plasmogamy, karyogamy occur, and then after karyogamy, that is the N negative fuses with the N negative strain. So the diploid nuclei is formed, and then diploid nuclei undergo meiosis. That is, it produces two negative strain, two positive strain, and then after meiosis, this type of resting structure with thick wall is formed. So this is how the structure. So this is the gametangia. These are the gametangia, and then after gametangia, this is the fusion, that is a zygosporangium, which contain a zygospore. In fact, zygosporangium, this zygosporangium, before it undergoes a period of rest, it undergoes meiosis. Meiosis or reduction division. Meiosis or reduction division. Reduction division. So, after reduction division, two negative strains and two positive strain nucleus is formed. Out of these, only one survive. Either it will be either a positive strain or a single negative strain nuclei. Just either this or this will be produced. So a single strain will be produced after meiosis. All the three disintegrate, three haploid nuclei will disintegrate, and then only one haploid nuclei will remain. That haploid nuclei, either this positive or this negative, so either any of this will again undergo mitosis. So there we see that in the zygosporangium, there is first meiosis, then there is disintegration of three haploid nuclei. Then one nuclei will survive, that will undergo mitosis, and they produce many spores. So this structure, this zygosporangium containing many these small zygospore, they are all of a same type, that is either positive strain or negative strain. So once this all the spores are mature enough, then there is disintegration, and these spores are liberated, which, if the substratum is suitable, they will germinate into a mycelium. So we see here there is also a certain bit of difference between reproduction in mucor as well as rhizopus. In rhizopus, this type of meiosis occurs after the dormancy, but here in mucor it occurs, this meiosis occurs before dormancy. So in rhizopus, two type of strains are produced, spores are produced, but in mucor only one type of strain of spore is produced inside this zygosporangium. So this zygospore, um, so this is a basic this zygospore how it looks. This is the zygospore, and this is how the sporangiospore looks. So there is a basic difference between zygospore and the sporangiospore. So zygospore is produced in sexual reproduction. It is produced by a sexual reproduction. All these spores, they are produced from sporangiospore. Sporangiospore. This is the sporangiospores, and this is how the zygospore looks like. So this is basically we see that the wall of the sporangiospore is much more smaller, and this wall is also more smooth. But in case of zygospore, that is this one, the wall is having various ornamentation. It looks very spiny and spiky in comparison to the sporangiospore. So though we stain the vegetative structure as well as reproductive structure with lactophenol cotton blue, so but from the shape itself, you can easily point out that which one is the zygospore and zygosporangium and the sporangiosporangium. So we can easily differentiate that whether the asexual or sexual type of reproduction we are seeing. When we are seeing a very spiny structure, then it is the sexual, that is the zygosporangium. And this when we are seeing a very smooth this structure, then it is a sexual reproduction. So now the life cycle, usually a mycelium, under when the condition is not that much drought prone, they produce this sporangiospore, which produces this the sporangium that produces this sporangiospore. And this sporangiospore, they if they fall on suitable substratum, they develop into a mycelium. And if the condition is not that much conductive, then what it produces? Then it produces this chlamydospores. So this chlamydospore, when the mycelium it dries away, disintegrate, these chlamydospore are liberated. They remain dormant, and on return of favorable condition, they what they do is they germinate into a mycelium. So this is how it occurs. This is the asexual reproduction. And then we have this sexual reproduction where opposite strain, say negative and positive strain, the two mycelium, when they come in close contact, when trisporic acid is liberated, and they produce this progametangia. This is the gametangia that I'm writing in PG. Then progametangia form the gametangia, and the suspensor cell here, this is the suspensor cell, this is the gametangia. And when these gametangia are mature enough, this wall, that is the wall in contact of the two opposite gametangia, they dissolve and fusion occur. And once the fusion occur, then after fusion they undergo meiosis. So first there is plasmogamy, then there is karyogamy, then there is meiosis, then there is either positive strain or negative strain single spore, because of the four haploid spore nuclei that is formed by the division of meiosis, only one nuclei survive. So either this or this, either positive or negative. So these again undergoes what? These undergo mitosis, and several nuclei of the same strain, that is either positive or negative, is produced. And these this sporangium contain many spore. So this is the sporangium, and um, so this is the germ sporangium. So the zygote, they germinate and produce the germ sporangium, which has many spores, and all these spores are either positive or negative, and these spore again germinate to produce this mycelium. So this is entire how the life cycle. So here we see that this stage, that is the zygospore stage, is only diploid. This stage is only diploid. Once the zygote, that is zygospore before this resting period, that is the when the zygote is formed, before this resting period itself, it undergoes mitosis. So only diploid stage is the zygo zygospore stage. Then once the zygote is formed after the after the dormancy, so there only we get only haploid cells. So only diploid stage is this stage. So this is how it differs from rhizopus. So that was all about the reproduction in Mucor, and I have made videos on other type of fungi also, and all I have put in the description box together in a playlist. You can go and watch those videos. And recently you have noticed that maybe I'm not making pure botany videos, but that is because I have been making videos on the GSET and NET question papers. You can also watch those videos. They are also very informative, and in fact that will help you in getting through NET and SET. So you can go and watch those videos also. And if you like this video, please do share it with your friends, and please do subscribe to my channel, because that encourages me. And I do make videos on pure botany topic also, so you can go and watch go to my channel and watch those videos. And if you have any suggestion or anything you want me to make videos on, please write it in the comment section. I will be making videos on those topic also one by one. And thanks again for watching this video.